Pharmapal.
Antifungal effects of Ambroxol Hydrochloride. Hey everyone! Welcome back to our channel. Today, we're diving into some fascinating research about the antifungal effects of Ambroxol Hydrochloride, especially its synergy with fluconazole against resistant strains of Candida albicans. Let's break it down. First off, what exactly is Ambroxol? Ambroxol hydrochloride is a medication primarily used as a mucolytic agent to help clear mucus from the respiratory tract. Recent studies have shown it may also have antifungal properties, making it a potential player in treating fungal infections. Now, let's get into the details of the study we're summarizing. The research focuses on the synergistic effects of Ambroxol when combined with fluconazole, an antifungal medication, against Candida albicans, particularly resistant strains. This study was published in Frontiers in Cellular and Infection Microbiology and involved both in vitro and in vivo experiments. Before we proceed, let's clarify some terms. In vitro, experiments conducted in a controlled environment outside of a living organism, like in petri dishes. In vivo, experiments conducted in living organisms, such as animal models. Synergism, when two substances work together to produce a greater effect than either would alone. Let's start with the in vitro findings. The combination of Ambroxol, ABH, and fluconazole, FLC, showed synergistic effects on resistant strains of C. albicans. The study used a method called the FIC index to measure this synergy, where values less than 0.5 indicate a strong synergistic effect. For instance, the minimum inhibitory concentration of fluconazole decreased significantly when combined with Ambroxol, showcasing its potential to enhance antifungal efficacy. Now, onto the in vivo results. The researchers used Galleria melanella larvae as a model organism to study the effects of the drug combination in a living system. Results indicated that the combination of ABH and FLC significantly improved the survival rates of infected larvae compared to those treated with either drug alone. Additionally, there was a notable reduction in the fungal burden, meaning fewer fungal cells were present in the larvae treated with both drugs. So, how does this combination work? The study suggests that Ambroxol may enhance the uptake of fluconazole while reducing its efflux, which means it helps the antifungal stay inside the fungal cells longer. This mechanism could potentially reverse the drug resistance seen in some Candida strains. Next, we'll explore the key tables and detail their experimental results. Table 1, Drug interactions of FLC and ABH against Candida spp. In vitro. First up, we have Table 1. This table summarizes the minimum inhibitory concentrations, MICs, of FLC and ABH, both alone and in combination, against various Candida isolates. What does MIC mean? It's the lowest concentration of a drug that prevents visible growth of a microorganism. The FICI model is also presented here, which helps us understand the interaction between the two drugs. A FICI value of less than 0.5 indicates synergism, meaning the drugs work better together than alone. Here's a quick look at some of the data. For resistant strains like CA10 and CA16, the combination of FLC and ABH significantly lowered the MIC from over 512 to just 2 g/ml. This is a clear indication of their synergistic effect. Table 2, Synergistic effects against biofilms. Next, let's move on to Table 2. This table assesses the synergistic effects of FLC and ABH against Candida biofilms at various stages of development, 4, 8, and 12 hours. Biofilms are clusters of microorganisms that stick to surfaces and are often more resistant to treatment. The SMIC, or sessile mic, is also listed, showing the antifungal concentrations needed to inhibit biofilm growth. The results here are impressive. The FICI values consistently show synergism at all time points, indicating that the combination of FLC and ABH is effective even against established biofilms. Mechanism of action, uptake and efflux studies. Now, let's talk about the mechanisms behind these results. The study also explored how ABH affects the uptake and efflux of rhodamine 6G, RH6G, in resistant Candida cells. Uptake refers to how much of the drug enters the cells, while efflux is how much is pumped out. The results showed that ABH significantly increased the uptake of Rh6G while decreasing its efflux, suggesting that ABH might help improve the effectiveness of FLC by enhancing drug retention within the cells. In vivo antifungal effects. Finally, the paper also discusses the in vivo effects of the drug combination using Galleria melanella larvae as a model organism. The survival rates of infected larvae treated with the combination of FLC and ABH were significantly higher compared to those treated with either drug alone. This is a promising indication that this treatment could be effective in real-world scenarios. In summary, the tables presented in this research provide compelling evidence that combining FLC with ABH can enhance antifungal activity against resistant Candida species, both in vitro and in vivo. Research figures. Before we delve into the figures, let's quickly summarize the key objectives of the study. Objective: to investigate the synergistic effects of Ambroxol Hydrochloride (ABH) and fluconazole (FLC) against resistant Candida albicans strains. Methods: the researchers employed both in vitro and in vivo experiments to assess the antifungal activity and mechanisms of action. Let's start with Figure 1, which illustrates the in vitro antifungal activity of ABH combined with FLC against various Candida isolates. Key terms. MIC, minimum inhibitory concentration, the lowest concentration of a drug that prevents visible growth of a microorganism. FICI, fractional inhibitory concentration index, used to determine the interaction between two drugs. In this figure, we see that the combination of ABH and FLC significantly reduces the MIC for resistant strains, indicating a strong synergistic effect. This is visually represented by the bars showing lower MIC values when both drugs are used together. Moving on to Figure 2, we focus on the effects of the drug combination on Candida biofilms. Biofilms, these are clusters of microorganisms that stick to surfaces and are encased in a protective layer, making them harder to treat. Here, the researchers demonstrate that the combination treatment not only reduces the fungal burden but also shows a marked improvement in survival rates of infected larvae, suggesting that this combination could be effective in clinical settings. Now, let's take a look at Figure 3, which presents the in vivo antifungal effects observed in Galleria melanella larvae models. Galleria melanella, this is a model organism used in research to study infections and immune responses. In this figure, we see a comparison of survival rates among larvae treated with FLC, ABH, and their combination. The results indicate that larvae receiving the drug combination had significantly higher survival rates compared to those treated with individual drugs. This highlights the potential of this combination therapy in enhancing treatment outcomes. To wrap things up, the combination of Ambroxol hydrochloride and fluconazole presents a promising strategy for overcoming antifungal resistance in Candida infections. This research opens new avenues for treatment options, especially for patients battling resistant fungal infections. Thanks for tuning in. If you found this video informative, don't forget to like, subscribe, and hit that notification bell for more updates on the latest in medical research. Let us know your thoughts in the comments below. Until next time, stay healthy.
Pharmapal.